For today, I will speak on behalf of the COVID human genetic effort, which I will bring into more detail a few minutes later. Because what I'm going to do is I'm going to tell you about our consortium efforts aiming to dissect this human genetic basis of resistance to SARS-CoV-2 infection. But in this particular context, I think it is important to realize that SARS-CoV-2 is not quite the first pandemic in human history. And in effect, even pandemics have been just with us throughout human history. Ironically, the Museum of the City of New York had a very interesting exhibition just prior to the start of the current pandemic. It was called Germ City. And very nicely, the curators also highlighted the breakthrough work throughout the last century by the major academic institutions in the city, amongst which Columbia, of course. Now, on a global and also historical scale, I believe everyone will be familiar with the various plagues that have swept throughout, particularly Europe, and that were listed by David Morris and Anthony Fauci in their recent perspective in Cell. But going even further back in human history, just prior to the current pandemic, it was shown that Neanderthal DNA introgression in humans provided adaptive means against viruses. And similarly, the study of ancient DNA has also provided proof for a viral epidemic in Southeast Asia millennia ago. And interestingly, most likely caused also by an ancient coronavirus. So what is important to us is that these pandemics, by means of pathogen-imposed selection pressures, that they may have been drivers of human adaptation, and they may have resulted in purifying and positive selection events for either advantageous or disadvantageous traits in humans. And this brings me to SARS-CoV-2, because one of the most remarkable features about this virus is the tremendous interindividual variability upon exposure in humans. And exposure may result in severe infections leading to death, while others develop only mild disease or are completely asymptomatic, while yet others might not get affected at all. In an attempt to understand this interindividual variability from the angle of human genetics, Jean-Laurent Casanova and Helen Su at the NIH early on in the pandemic initiated the COVID human genetic effort, which is now a global consortium with over 150 collaborators and more than 50 sequencing hubs worldwide. And today I will walk you through our consortium's genetic and immunological work on the various extreme phenotypes of COVID as a lineup to tell a bit more on our most recent quest to understand the human genetic basis of resistance to SARS-CoV-2. So our first problem to crack or phenotype, so to say, was what actually causes critical COVID pneumonia. Because upon infection, only a very small minority of individuals infected develops a critical disease, while the vast majority has only mild symptoms or perhaps no symptoms at all. So while there were a few epidemiological risk factors quickly identified, like sex, age, and other comorbidities, there's still an inter-individual clinical variability remaining within these demographic groups. So the consortium set out to identify the actual causes of lethal COVID pneumonia. And here we took lessons from other viral infections like influenza, where the involved labs had identified inborn errors of type 1 interferon immunity in a few individuals suffering from severe respiratory viral diseases prior to the pandemic. Indeed, we identified various inborn errors of type 1 interferon immunity in about 3% of patients with life-threatening COVID pneumonia from across the continents when we made a comparison to silent or mild COVID as controls. And surprisingly, most of these patients with type 1 interferon immune deficiencies, they had been completely fine up to COVID, indicating incomplete penetrance, also for other viral diseases like influenza, but also a level of redundancy of type 1 interferons. In a follow-up study later on, we identified X-linked recessive TLR7 deficiency as a very penetrant genetic etiology of critical COVID pneumonia, where about 1.5% of men under 60 years of age with critical COVID are TLR7 deficient, suggesting that human TLR7 and PDCs are essential for the host's defense upon infection with SARS-CoV-2. So together, the identification of these inborn errors of type 1 immunity has implications for the diagnosis and screening in patients with severe COVID pneumonia, but also for the prevention and treatment with interferons in these patients, perhaps. At the same time, and driven by our identification of 3 APS1 patients, which is an autoimmune disorder, amongst these critical COVID pneumonia cases, we found that about 10% of critical COVID-19 patients had autoantibodies against these very same type 1 interferons. And these autoantibodies, they were pre-existing and neutralizing. Over 90% of these patients were men, and half of them over the age of 50 years. And the autoantibodies are just very rarely present in asymptomatically infected individuals or healthy controls. And this initial study was, in the meanwhile, replicated by various others across the globe. Now, in a follow-up study, we found that these autoimmune phenocopies of inborn errors of type 1 interferons that are even more prevalent at lower concentrations of type 1 interferons, showing here in the left lower part of the slide. And that collectively, they can account for up to 20% of COVID deaths at all ages. And by screening really thousands of pre-pandemic sera from at that time healthy subjects, we're also able to identify an age-dependent risk, where the current estimate is that about 4% of individuals older than 70 years of age has these autoantibodies against type 1 interferons, which is shown in the right lower part of the slide. Now, these findings indicate that critical COVID-19 can be caused by autoimmunity against intrinsic immunity, and that's similar to the inborn errors of type 1 interferon immunity. The identification of these autoantibodies might have implications, again, for diagnosis, prevention, and treatment. So inborn errors of type 1 interferons, including autosomal TLR3 and X-linked TLR7 deficiencies, underlie critical pneumonia in at least 5% of patients overall, and pre-existing neutralizing autoantibodies underlie critical pneumonia in at least another 15% of patients. So taken together, about 20% of cases with critical COVID pneumonia can now be explained with the inborn errors giving an age-independent baseline risk and the autoantibodies an age-dependent risk, in particularly in those over 70 years old. And biologically, critical COVID pneumonia seems to revolve around defects of 2 cell types and 1 set of molecules. So the TLR3 dependent production of type 1 interferons by respiratory epithelial cells, and the TLR7 dependent production of these cytokines by PDCs, which are both crucial for host defense against SARS-CoV-2. And in pathophysiological terms, these genetic and biological data suggest a 2-step model where insufficient type 1 interferon immunity in the respiratory tract during the first few days of infection accounts for the subsequent spread of the virus, leading to pulmonary systemic inflammation in this second step. So the base camp for the impact of human genetics on the interindividual variability in the course of COVID-19 seems to be strong. And now that we had cracked the first COVID phenotype, we have also engaged in investigating the human genetic and immunological causes of other COVID-related phenotypes, like the pediatric syndrome MIS-C, COVID-TOS, and natural resistance to infection with the virus. So, for instance, the consortium has recruited a cohort of about now 750 pediatric patients with a multisystem inflammatory disorder. And we suspect at least some of these cases might be genetic, also given similarities of the disorder to traditional Kawasaki disease. The genetic analysis and mechanistic characterization are currently underway. And similarly, we have recruited about 300 individuals with pandemic-associated pernio. better known as chilblains or COVID toes, including some multiplex families like the one showed here. The clinical manifestations and also histological findings again suggest similarities with type 1 interferonopathies. And also here, the genetic investigations are ongoing at this time. But last but not least, the phenotype and the major topic for today is about possible natural resistance to SARS-CoV-2. And the idea to initiate a study into the human genetic and immunological basis of resistance to SARS-CoV-2 infection was again born from the interindividual variability in the outcome upon exposure to the virus. And here it seems that a few people, apparently against all the odds, like being at age and having multiple and serious comorbidities, simply do not get infected despite an intense and repeated exposure to SARS-CoV-2. So for instance, we came across this elderly lady who took care for her husband during the days preceding his ICU admission in the early March first wave, while she had not had access to face mask or other PPEs at that time, or the non-engineering living in a skilled nursing facility that was affected by a devastating outbreak of infections during this very same first wave, where this guy ended up being the only one in his ward not being affected, or a mother in whose household all the family members got infected except of her, despite she had regular and of course unprotected interactions with all of them, including her husband and the kids. And for all these exceptions, both PCR and serological tests have remained negative repeatedly. So there's of course multiple scenarios as to why these people would not get infected. And one of these is that prior acquired cross-reactive adaptive immunity could block the infection at a very early stage. And a recent study in seronegative healthcare workers indeed suggests a role in some people for pre-existing memory T-cell responses in supporting the viral control and the early abortion of the infection. But another scenario is that inborn or genetic defects could confer an intrinsic immunity to the infection that is specific to SARS-CoV-2. And as a matter of fact, there's a few historical examples in support of this scenario, at least in some individuals. I will come back to that in a few slides. So our focus is to test the hypothesis that some of these cases of natural resistance to infection with SARS-CoV-2 may be genetic. But there's a subtle, but we think important conceptual difference between protection and resistance. Because protection against infection typically results in a small individual effect size when observed as a group by means of a relatively weak biological effect. But then in contrast, the resistance to infection that can provide a large individual effect size based on a very strong biological effect. And our consortium aims for this last category. And again, there's a couple of historical examples that go in support. Because as early as in the 1970s, Louis Miller, he discovered that the absence of the Duffy antigen on erythrocytes prevented these cells from becoming infected with Plasmodium vivax. And it is now clear that this trait was positively selected for in malaria endemic areas across time. But the molecular genetic basis of the autosomal recessive resistance trait was only discovered in the 1990s. And the causal variant affects the GATA1 binding site in the dark promoter, selectively preventing the gene transcription of this gene in erythrocytes. At about the same time, autosomal recessive CCR5 deficiency was found to confer resistance to infection with HIV-1. And the most common loss of function mutation in CCR5 is deletion, with a minor allele frequency of about 10% in the European population, for which the evolutionary driving forces are not really known at this time. And then finally, there's autosomal recessive FAT2 deficiency, which was discovered to confer resistance to gastrointestinal infections with norovirus. And this is the so-called non-secretor type, which is affecting the H antigen. So these historic examples of Mendelian resistance to infection have three things in common. First, all three are complete deficiencies. Second, they affect the receptors or co-receptors exploited by the pathogen as a means of entering the cells. And third, they are clear drug targets for the treatment of infection in others who are not deficient. So what does this all mean for SARS-CoV-2 and for candidate genes for natural resistance to infection? And interestingly, at this time, and despite a good 2 years in the pandemic, we do not precisely know the proportion of humans that are actually naturally resistant to infection. Nevertheless, a number of candidate genes have emerged from multiple lines of evidence. And one is the ABO locus, which was identified in GWAS studies. And a meta-analysis has indeed confirmed an effect of this locus on susceptibility to infection, where the protective effect of the O allele is, however, quite small, with an odds ratio of around 0.9. To be discovered genetic etiology of COVID-TOS, possibly related to interferonopathies, could also provide insights into the mechanisms of resistance to infection, as these could teach us what the host mechanisms are that restrict viral replication and actually promote resilience upon infection with SARS-CoV-2. Now, another line of evidence comes from the various interactome studies, which shed light on the viral life cycle upon infection. And here, obviously, the receptor ACE2 comes into the picture, but also other related genes like TMPRSS2. And interestingly, NOMAD reports 2 male individuals carrying essential splicing site mutations. And of course, we would love to get to know these men. Now, we envision 2 general biologic mechanisms causing resistance to infection. The first mechanism would result in unpermissiveness for viral binding or entry to the host cells, while the other would result in an immediate abrogation of the infection by not permitting subsequent viral replication. But both ultimately could result in resistance to SARS-CoV-2 infection. There's 2 key challenges in the search for individuals who may be naturally resistant to infection with SARS-CoV-2. And the first challenge lies in the probability of virus transmission upon exposure. And this likelihood is influenced by both the duration and the intensity of exposure to the infected individual, and the intrinsic transmission characteristics of the pathogen, which, as we all know, is dynamic and dependent on the variant. So for our approach, we focus on household contacts and preferably bed partners who have had a prolonged and unprotected close interaction, meaning for several hours a day during several days without using PPE, with a symptomatically infected individual who ended up either being admitted to the hospital or at least requiring assistance with daily activities, meaning a score higher of 3 on the WHO clinical progression scale, as was just mentioned also in the previous talk. Then the second challenge lies in diagnostically demonstrating the absence of an infection, since PCR only provides a snapshot information and some asymptomatically infected individuals might remain seronegative. We will also assess SARS-CoV-2 specific T-cell responses after the enrollment of all our individuals. And then, in order to pick up potentially rare variants with a large individual and biological effect size, we will perform whole exome and whole genome sequencing, and then compare the candidate resistors to various other COVID-19 phenotypes that we have here in-house. And then finally, to dissect the mechanisms of resistance, the genetic findings will be validated experimentally, including in primary patient samples, which is essentially very similar to all the other work by the COVID human genetic efforts. So we wrote about this project in Nature Immunology earlier this summer, and to our surprise, actually, our perspective gained a lot of traction in the scientific community. It was highlighted by Nature, but also by the general public with a lot of media attention. So consequently, our monthly website visits have literally exploded. And in only a few weeks' time, we have now received a good 5,000 responses from individuals from across the globe who recognize themselves and who would love to participate in our studies. So we have set up quickly an online and now multilingual pre-screening survey in order to validate their eligibility. And we are currently in the process of recruiting these additional candidate resistors who come on top of the nearly 650 that we had already enrolled. So our enrollment is ongoing, and of course, the more the merrier for this study. So we will really warmly welcome new participants. So in the event that you would like to team up, please don't hesitate to reach out. And with that call for your help, I'd like to wrap up, but not before acknowledging everyone involved. In particular, Jean-Laurent and Helen, and Laurent, but also Evangelos Andreas, who are the key players in this project. But also Yelena Nemirovskaya, with whom it would not have been possible to handle the absolutely overwhelming number of responses from the general public. And of course, all the other members of our consortium. So with that, I would like to thank you for your attention, and I'll be happy to answer your questions.
Thank you, Andreas. If I could start out, and I realize you can't answer this yet, but maybe just get your thoughts. Can you comment on how common you think inborn resistance is going to be? And I ask this because, you know, we all know people who've been infected, and I know people who, you know, one family member or multiple family members get infected and then others do not, and they're always asking me why, and I can't answer them. So any thoughts you may have on that would be great.
Yeah, that's a very important question that you raise, and this is also a question that at this time we are not so sure about precisely. So to start with, I think it is important to realize that there might be multiple mechanisms at play that might result in the very same not being affected situation for a household contact. And what we aim for is to identify what the genetic mechanisms could be, at least in some of these individuals. And we take into account that there might be multiple frequencies actually at play of minor allele frequencies that could define these traits. What we know is that the secondary attack rates, it's actually quite variable within households. In some households, like, you know, for Omicron, this might be different, but for the prior variants, the secondary attack rates varied in between 30 to about 80% in households. So that could suggest that such a phenotype would have a prevalence of about 20%. However, it's not being said that all those 20% are being explained by inborn genetic traits. So we take into account that these traits might be very, very rare. And I think one of the examples is also ACE2, for which we already know, based on population genetics, that deficiency is going to be ultra rare based on what is reported in large public databases. So we do take into account that these traits are extremely rare, and that is also what makes our approach different from genome-wide association studies, because our technical strategy allows us to identify these extremely rare cases, to pick them up and subsequently investigate their biological meanings.
Perfect, thank you. We have a question from Rodney Rothstein in the audience. Rodney, you should be able to ask your question now.
Yes, thanks for that very interesting talk. I was curious about whether you're only doing genetics or whether you're also thinking about doing metabolomics and actually asking something about many different aspects of the patient rather than just their genes, if you understand exactly what I mean.
Yeah, so again, yet another great question. And what you are asking is also, you know, the various different drivers that could result in the endpoint. And in that way, indeed, we do take into account that there might be other causes in particular individuals. But again, what we aim for is to identify, at least in some of those people, what might be their inborn mechanism of resistance to SARS-CoV-2. There might be indeed other mechanisms that play in yet other people. The metabolomics studies, that is beyond what we aim for at this time. Also, given the strong historical examples that we have from other endemic diseases like malaria, the noroviruses, and HIV, and the historical proof of concepts actually that this indeed can be the case.
Thanks.
Okay, we have a question from Zhizheng Sheng. You should be able to talk, Zhizheng.
Hi, very nice talk. I have a question about the autoantibody. You mentioned before for some of the patients with symptoms, there's a pre-existing type 1 interferon autoantibodies. Could you expand a little bit on whether there are inborn or other genetic factors contributing to the autoantibodies in the patients?
Yes, thank you again. Thank you for raising this question. So your question is, what is causing these autoantibodies in these people? If I take it well. And the answer there is that at this time, we do not yet know the cause. However, these autoantibodies in themselves, the autoimmunity, is a genetic disease in itself. And the $1 million question is what is the genetic cause here? And we are putting a lot of efforts in identifying the cause here at this time, but we don't know yet.
You think there's not, like, for example, environmental factors like infection resulting autoantibodies in this case? It's basically genetic.
Yeah, so we have good indications that these autoantibodies in themselves are driven by genetic causes. And the reason why we think is this is also given the fact that we came on this track for the autoantibodies by the identification of 3 APS1 deficient patients in our initial cohort of severely ill COVID-19 patients. And this put us on a track in identifying autoantibodies in a whole lot of additional patients that were not necessarily APS1 deficient, but that seem to be connected.
Thank you.
Okay, we have one more question from the audience before we move on, and that's from Francis. Sorry, I can't see your last name here. Akita?
That's right. I just have a question. I would like you to comment on the effect of the Neanderthal gene and the fact that it makes people prone to have severe infection.
Yes, so the Neanderthal introgression, that is an interesting that's an interesting topic. Just as an anecdote, I received an email 2 days back from someone somewhere in the US who had read about our work and who was forwarding her commercial genetic whole exome sequencing information, and she proudly presented the fact that she overrepresented Neanderthal DNA compared to other people, and what we thought about that. So, the Neanderthal DNA introgression, there is a clear effect. However, the effect is quite modest, actually, in terms of the individual impact. So on a very big population, you can pick it up, right? Because then there is statistics that you can drive on. But for the individual, the biological impact is very, very modest. And this is also what our approach differs from. We identify perhaps very rare variants that have a big individual impact.